Hello everyone, I welcome you all in the lecture series on biomolecules. Today is our 11th lecture, and before going to the details of today's lecture, I will like to give a recap of my last lecture. In the last lecture, we talked about enzymes and how do they function, basically how catalysis takes place, and what are the basically what type of functional groups are there at the active side of the enzyme which are responsible for the catalysis. And then I discussed little bit of vitamins, and in vitamins I talked about the type of vitamins. Basically there are two types of vitamins, water soluble vitamins and water insoluble vitamins. So, continuing with the same, where did I leave that basically I was mentioning that vitamin C and vitamin E are radical inhibitors and therefore are antioxidants, and vitamin C basically traps radicals. Vitamin C traps radicals formed in aqueous environment, formed in aqueous environment, whereas vitamin E traps radical vitamin E traps radicals formed in nonpolar environment. In nonpolar environments. So, now I will go in the details of this water soluble vitamins and water insoluble vitamins, that how many vitamins are water soluble and what is their source, and their deficiency leads to what kind of disease. These are the things I want to explain you one by one, so that we can learn more about these biomolecules. So, let us talk first about water soluble vitamins. Water soluble vitamins. So, first water soluble vitamin is, first water soluble vitamin is thiamine, vitamin B1. Thiamine, which is vitamin B1. And what is its source? Its source is yeast, milk, green vegetables, green veggies basically, and cereals. These are the sources, and its deficiency leads to very disease. Very disease. What is very disease? Loss of appetite. In very disease, what happen? Loss of appetite, and retarded growth that is happening, retarded growth. Retarded growth. Second water soluble vitamin is vitamin B2. Vitamin B2, and vitamin B2 is also known as riboflavin. Riboflavin. Riboflavin and vitamin B2 can be obtained from milk, egg white, liver, kidney. And its deficiency leads to cheolosis, What is cheolosis? Cheolosis leads to the fissuring of corners at corners of mouth and lips. Mouth and lips. So, this is basically cheolosis. In cheolosis, you will see that fissuring at corners of mouth and lips occurs. Now, the third vitamin is vitamin B6. Vitamin B6, which is also known as pyridoxine. Pyridoxine is obtained from yeast. So, you can see that yeast, if you take yeast in your dietary content, then you can compensate thiamine as well as pyridoxine, vitamin B1 and vitamin B6. Is milk. Milk is another dietary intake through which you are compensating all the three vitamins. So, milk is very important for our dietary intake. Egg yolk, cereals and grams. And grams. And deficiency of vitamin B6 leads to the convolutions Now, the fourth vitamin, water soluble vitamin, is vitamin B12. Vitamin B12, and vitamin B12 can be obtained from meat, fish, egg and curd. Deficiency of vitamin B12 leads to pernicious anemia. pernicious anemia. And basically what happens there, that RBC deficiency causes in hemoglobin, RBC deficient in hemoglobin. In hemoglobin. Now, I will come to the fifth vitamin. Fifth vitamin is vitamin C. As I have already mentioned that vitamin C is required in fairly good amount, and it should not be called vitamin, because when I was defining vitamin, I told that vitamin is basically required in a small amount, whereas vitamin C I told you that it's required in fairly good amount, and this can be synthesized also inside the body. However, it can be also obtained by the dietary intake, vitamin C. Vitamin C, which is also known as ascorbic acid. Ascorbic acid. Ascorbic acid. Vitamin C can be obtained from citrus fruits. Citrus fruits such as you can get it in amla and green leafy vegetables. Amla and green leafy vegetables. And deficiency of vitamin C leads to the scurvy disease actually. Scurvy, and they are basically bleeding. The symptom is that bleeding gums, bleeding gums. Now, we will talk about water insoluble vitamins. Water insoluble vitamins. Water insoluble vitamins. What are water insoluble vitamins? Vitamin A is water insoluble vitamin, and this can be obtained from fish liver oil, carrots, butter and milk. Butter and milk. And deficiency of vitamin A leads to xerothalmia. Xerothalmia. Basically, what happens here? Hardening of cornea of eye. Hardening of cornea of eye. Cornea of eye. Another vitamin is water insoluble vitamin is vitamin D. Vitamin D, and vitamin D, nowadays vitamin D deficiency is very frequent, particularly in people who are working in offices, because we do not expose in sufficient amount to our body to the sunlight, and for the formation of vitamin D, sunlight is very, very important. If you want to synthesize vitamin D inside your body, vitamin D exposure to sunlight fish and egg yolk. Fish and egg yolk. Deficiency of vitamin D leads to rickets, rickets basically leads to, ricket disease is, kind of leads to the bone deformities in children. Bone deformities in children and osteomalacia. Soft bones and joint pain in adults. Soft bones and joint pain in adults. Joint pain in adults. Deficiency of vitamin D leads to these problems. Third one is vitamin E. Vitamin E. Vitamin E can be obtained from the vegetable oils. Vegetable oils like wheat, germ oil, sunflower oil, etc. And deficiency of vitamin E leads to the increased fragility of RBC. Increased fragility of RBCs and muscular weakness. Muscular weakness. And finally, vitamin K can be obtained from green leafy vegetables. So, green leafy vegetables also we saw that can supplement to these water insoluble vitamins. That is also very important for the having good balance of vitamins in our body. So, it is always advisable that one should take a lot of leafy vegetables in their dietary intake. So, vitamin K can be maintained by green leafy vegetables. And deficiency of vitamin K leads to the increased blood clotting time. Increased blood clotting time. blood clotting time. So, this much about the vitamins. Again I will like to repeat vitamins classification. So, water soluble vitamins, first one was thiamine or vitamin B1, and vitamin B1 can be obtained from yeast, milk and green vegetables and cereals. Deficiency of vitamin B1 leads to the beriberi disease, where basically symptoms are like loss of appetite and retarded growth. Then next one is vitamin B2 in the water soluble vitamins, and it can be obtained from milk, egg white and liver and kidney. Deficiency of vitamin B2 or riboflavin leads to the chelosis basically, and in chelosis fissuring at corners of mouth and lips, these are the symptom. Vitamin B6 or pyridoxine can also be obtained from yeast, milk, egg yolk, cereals and grams, and deficiency of vitamin B6 or pyridoxine leads to the convulsions. Vitamin B12 can be obtained from meat, fish, egg and curd, and deficiency leads to the pernicious anemia, where RBC deficient hemoglobin basically is the reason. Now, the fifth one is vitamin C or ascorbic acid, water soluble vitamin, and that can be obtained from the citrus fruit, amla and green leafy vegetables, and deficiency of this leads to the scurvy disease, and that is symptom is bleeding gums. And this much about the water soluble vitamins. Water insoluble vitamins, in that series vitamin A, and vitamin A can be obtained from fish liver oil, carrots, butter and milk, and deficiency leads to the xerophthalmia, where hardening of cornea of eye is the symptom. And vitamin D in the water insoluble vitamin is can be maintained by the exposure to sunlight. If we expose our body to sunlight, then that way we can maintain proper level of vitamin D, and it can also be maintained by fish and egg yolk. Deficiency leads to the rickets, bone deformities in children, and osteomalacia, where basically soft bone and joint pain, these are the symptom of vitamin D deficiency. And as I mentioned that it is quite common in the people who are working in the offices, like who are not getting proper exposure of sunlight to their body actually. And particularly in the metros, because of the nature of job, a lot of population is getting affected with the vitamin D deficiency. Then the third one is vitamin E, and vitamin E can be maintained by the dietary intake of vegetable oils like wheat germ oil and then sunflower oil, and deficiency leads to the increased fragility of RBCs and muscular weakness. And the fourth one is in the water insoluble vitamin is vitamin K, which can be obtained from green leafy vegetables, and deficiency leads to the increased blood clotting time. Basically, vitamin K helps in the clotting of blood. Whenever we get any wound, in that we always require to clot that brucus. Otherwise, if the bleeding keep on continue, then we will lose a lot of blood, and that there vitamin K helps because it immediately clot that, and it starts basically in healing, wound healing process. So, these are the role of vitamins. This much about the vitamins, and now I will continue the final biomolecule, nucleic acid. So, let us talk about the nucleic acids. Nucleic acids. Nucleic acids, how do you define nucleic acids? Nucleic acids are another biomolecules, and they are very important, because when we talk about in detail, then they are the our hereditary materials basically. And let us learn about what is the structure of these hereditary compounds, what kind of molecules, and what kind of functional groups are available there in the nucleic acid. So, how can we define nucleic acid? So, nucleic acids are chains of 5-membered ring sugar, 5-membered ring sugars, linked by phosphate groups. Linked by phosphate groups. Notice that linkages are phosphodiesters. Here in the nucleic acid, notice that linkages are phosphodiesters. Phosphodiesters. In RNA, the 5-membered ring sugar is, what is the difference? So, we know that there are two types of nucleic acid, DNA and RNA. How are they different? So, in RNA, the 5-membered ring sugar is D-ribose. Five-membered ring sugar is D-ribose. Ring sugar is D-ribose. D-ribose. In DNA, the 5-membered ring sugar is 2-deoxy-D-ribose. Ring sugar is is 2-deoxy-D-ribose. D-ribose. Now, let us talk about the bases in DNA and RNA. So, nucleic acid, how to define nucleic acid? Nucleic acids are chains of 5-membered ring sugars linked by phosphate groups. Notice that linkages are phosphodiesters. So, every 5-membered sugar is linked by phosphate. In between two 5-membered sugar, there will be phosphate linkage. So, if you are having three sugar, 5-membered sugar, so there will be two phosphate linkages. So, that is why I told that, notice that linkages are phosphodiesters. In RNA, the 5-membered ring sugar is D-ribose, whereas in the DNA, 5-membered ring sugar is 2-deoxy-D-ribose, 2-deoxy without the oxygen. In the RNA with the regular actually D ribose, but in the DNA sugar is 2-deoxy, oxygen is not there, that is different, 2-oxy D ribose. Now, let us learn in detail about the DNA's and RNA's structure. So, the bases in DNA and RNA. Bases in DNA and RNA. I will like to draw the structure of DNA and RNA. So, as I was mentioning that phosphodiester, what is the phosphodiester? This is the phosphodiester linkage, and as I mentioned that here base is attached. Base is attached here, base, and again to I will not So, as I mentioned that RNA has D ribose. So, I will make the D ribose structure again, and this is the another phosphodiester linkage. Base again here, OH, and again this is the continuation part. In RNA, as I mentioned that at the 2 position it will have hydroxyl group. This is it has D ribose. So, 2 prime OH group, and this is the phosphodiester linkage. This is phosphodiester, a phosphodiester link. Phosphodiester links one 5-membered sugar to the next. One 5-membered sugar to the next. One 5-member to the next. Okay. And so, we can represent a phosphodiester linkage as if I put this D ribose with the R, and then again it is through the oxygen it is attached. So, and then this is phosphorus, and this O minus I can put it in the OH, and then again R. This is the another ribose, D ribose. This is one D ribose, this is another, and in between this is. So, this is basically a phosphodiester. This is called phosphodiester. Phosphodiester, and basically it is almost similar to the phosphoric acid, phosphoric. So, in the phosphoric what do we have? HO 3OS S3PO4. This is phosphoric, this phosphoric. So, this is about the RNA. Now, I will make a structure of DNA. DNA is also similar, but slightly different, as I told you that at the in the 5 membered ring sugar at the 2 position, it does not have hydroxyl group. So, here again it is attached with the base, it is attached with the base, attached with the base. So, this is RNA, this is RNA and this is DNA. Now, I will compare that in the DNA no 2 prime OH group, no 2 prime OH group, and also both D-ribose in the RNA and 2-deoxy D-ribose in DNA, both are having beta glycosidic linkage with the base. So, this is a beta glycosidic linkage. Beta glycosidic linkage. Here also it has a beta glycosidic linkage. Beta glycosidic linkage. So, this is the structural backbone for DNA and RNA. So, in what is the basic difference between the DNA and RNA is that sugar that which is part of its structure scaffold. In DNA it has 2-deoxy D-ribose 5 membered ring sugar, whereas in RNA it has D-ribose. And what other differences are there? The other differences are there in the base, what kind of base RNA is having and what kind of base DNA is having. Similarity is that both of them are linked with the phosphodiester linkages, which I showed in, that is common. In DNA also 2 5 membered sugars are connected with phosphodiester linkages, and in RNA also 2 5 membered sugars are connected with the phosphodiester linkages. So, as I mentioned that there are also difference in the bases available there. What kind of bases are there? There are only 4 bases in DNA, in DNA 2 are substituted purines, 2 are substituted purines. What are they? Substituted purines are adenine and guanine. Adenine and guanine, and 2 are substituted pyrimidines. Two are substituted pyrimidines. And they are substituted pyrimidines are cytosine and thymine. Cytosine and thymine. Cytosine and thymine. So, let me first introduce what is purine and what is pyrimidine. So, I will like to draw the structure of purine base. Purine base has 6 and 5 membered fused ring. So, I have drawn here 6 membered and then this is 5 membered. This is purine. How to number it? 1, 2, 3, 4, 5, 6, 7, 8 and 9. I told you that DNA has 2 substituted purines, adenine and guanine. So, I will like to draw the structure of adenine and guanine. Adenine, this is adenine, and basically in adenine it has at the 6th position amino group, and guanine guanine. So, adenine and guanine, and then pyrimidines, 2 substituted pyrimidines. So, let me write the structure of pyrimidine. Pyrimidine has a single 6 membered ring with 2 nitrogen. And again it can be numbered from 1, 2, 3, 4, 5 and 6. So, this is pyrimidine. So, DNA has 2 substituted pyrimidines, cytosine and thymine. So, let me write the structure of cytosine. And thymine is this is thymine. So, as I mentioned in the beginning that DNA has 2 purine bases, adenine and guanine, and it has 2 pyrimidine substituted pyrimidines, cytosine and thymine. Similarly, RNA also contains 4 bases. RNA also contains only 4 bases, three adenine, guanine and cytosine. 3 adenine, guanine and cytosine. Adenine, guanine and cytosine are same as those in DNA. Are same as those in DNA. The fourth base in RNA is uracil instead of thymine. The fourth base in RNA is uracil instead of thymine, is uracil instead of thymine. Instead of thymine. Let me draw the structure of uracil, how uracil looks like. So, this is uracil. Uracil. So, I will again like to rephrase about the structure of nucleic acids. RNA has D-ribose sugar, and these D-ribose sugar are linked with each other by phosphodiester linkages, whereas DNA has 2-deoxy-D-ribose sugar and these 2-deoxy-D-ribose sugar, this 5-membered ring sugar are linked with each other again with the phosphodiester linkages, and they have these sugars are having beta glycosidic linkage with the bases. DNA has 4 base, 2 basically 2 pyrimidine substituted pyrimidines and 2 substituted purines. Sorry, DNA has 2 substituted purines, adenine and guanine, and it has 2 substituted pyrimidines, cytosine and thymine. Here is the structure of purine bases. Basically, it has adenine has 6 amino purine basically, and guanine it has like oxo group at the 6, and at the 2 position it has amino group in the purine, and pyrimidines in the DNA it is cytosine and thymine. Basically, it is a substituted pyrimidines. RNA also contains 4 bases, and 3 out of 3 adenine, guanine and cytosine are same as those in the DNA. The 4th base in RNA is uracil instead of thymine. Here is the structure of uracil. So, this much about the basic structure of nucleic acid. Now, I will talk about the how these biomolecules are formed actually. So, first I will talk about the nucleosides. Nucleosides. What is nucleosides? A compound containing a base a base bonded to D ribose, bonded to D ribose or 2-deoxy D ribose, 2-deoxy or 2-deoxy D ribose is called a nucleoside. Is called a nucleoside. So, nucleoside in RNA, what are the nucleoside in RNA? Nucleosides in RNA. Nucleosides in RNA are, since we had the 4 bases, nucleosides in RNA will be base dependent. So, here D ribose connected with the adenine will be adenosine. Adenosine, D ribose connected with the guanine, guanosine. D ribose connected with the cytosine, cytidine. D ribose connected with the uracil is uridine. So, these are the nucleosides. I will stop here and I will again continue structure of nucleosides in the next class. Thank you very much for the attention.